Welcome to Unit 2. This is going to be the living world and the biodiversity section of the living world. So as you can see here, this is from XKCD. As far as land mammals by weight, which is essentially by mass, humans and all our pets and all our livestock make up an overwhelming proportion of the mass of mammals on land. Obviously, there are other animals, insects, reptiles, and so forth. But as far as mammals go, we and our livestock and pets pretty much dominate. So biodiversity at a very basic level is how much different life there is on Earth. And we're going to look at different ways to measure this. Three primary ways, and we'll talk about each of them. First is genetic diversity. So when you look at a population of individuals, genetic diversity is going to be the variation within that population. And genetic diversity may shift based on environmental stressors, right? Physical stress can cause changes in genetic diversity. Wildfires can cause changes, right? Certain individuals are eliminated and basically randomly. Pollution is an environmental stressor. Thermal stress, right? If things are too hot or too cold. Radiation, like UV radiation from the sun. Any other combination of climate conditions, amount of light and temperature. And finally, biological stressors, predation, competition, parasitism. So, genetic mutations arise randomly, but then which mutations survive depend on what stressors are out there. So consider this. What would enable a population to have more resilience to an environmental stressor? And you could consider that humans are experiencing this in 2020. So in your notes, try to come up with an example. So one effect we see sometimes with environmental stressors is that you have an original population, here represented by gumballs. They're primarily yellow. But if there's a stressor that basically bottlenecks the genetics, and only certain individuals survive, but those surviving individuals have a different genetic makeup than the original population, the new population will have a different gene pool. So you can see this certainly with extinction events and other random environmental stressors. The second type of diversity is species diversity. So this is how many different species are in a region or a habitat, right? So we're looking at the community or the ecosystem level. So we don't really know how many species there are, but we know what makes a species, right? We know that they have to produce viable offspring. We've cataloged 2 million species, but we know there are more out there. Every time somebody goes to the Amazon, they find new species that they haven't seen before. Even though rainforest is shrinking, there's still lots of areas that are new to scientists. And so some estimates for the total number of species, 5 to 100 million, but 100 million is on the high side. Most often people are thinking it's probably around 10 million. So most common animal on Earth, you might think it's a beetle or something, but it turns out that it is roundworms, nematodes, that they make up as much as 4 out of 5 animals on Earth. And so if you eliminated all the solid materials, you'd see a lot of nematode worms left over. But that's just the animal kingdom. If you look at all organisms, bacteria are the most common. This particular one, Pelagibacter ubique, makes up 1/3 of all single-celled ocean organisms. So after genetic diversity and species diversity, There's habitat or ecosystem diversity. So the College Board calls it habitat diversity, but our textbook calls it ecosystem diversity. For our purposes, those are the same things. And this is the variety of habitats that exist in a region. So if you take a look at this map of California, you can see lots of different habitats, and some of them look similar to biome names, but these are more detailed, right? You can have lots of different habitats within a biome. So you can see in California, they have quite a few different habitats. One thing that makes species able to diversify are whether they're generalists or specialists. So generalist species live in a wide range of conditions or habitats. A lot of omnivores fall into this category. A lot of primary consumers fall into this category. Basically, the fact that they can eat lots of different things makes it possible for them to survive varying conditions. Specialist species, on the other hand, have a very narrow range of conditions that they can live in, right? Koalas only eat eucalyptus, and pandas only eat bamboo. Amphibians are, surprisingly narrow, considering that they can live in air or water, but they're very sensitive to changing conditions. Meanwhile, this is a good point in the video to pause, and you can check out YouTube for cute baby panda videos. There are lots of cute baby panda videos, so check it out whenever you feel like it. So some more questions for thoughts for your notes. Consider what habitat loss would do to generalist species versus a specialist species. And in general, do you think specialist or generalist species are more likely to be in danger of extinction? However, why would it be advantageous to be a specialist as compared to a generalist? And then in terms of large or small territories, right, where generalists might have large territories, specialists might have small territories, what would habitat loss have on animals in either of these categories? So another way we can measure diversity in terms of species richness. Species richness is the total number of species, and so that's certainly helpful. Of course, that doesn't take into account how many individuals there are. So another measure we have of biodiversity is species evenness, and that's the abundance of individuals within each species. So let's take a look at an example of richness versus evenness. So consider these two forests, right? Community 1 and Community 2 both have the same number of species, right? They're both species rich in theory, but in terms of species evenness, they're not both even, right? So Community 1 is very even, whereas Community 2 is not even. Consider again some decomposers, right? Community A, the yellow mushrooms are relatively abundant, and so they are low evenness, even though they're species rich with 4 different species. Community B has the same number of species, so they are also species rich, but they also have a high evenness, which makes it a little more healthy. So there are 2 mathematical ways to measure species diversity, and we're going to use one of these in the parking lot lab that we're going to do. So the Shannon-Weiner diversity function, this looks like a mess, but you're going to get to practice with it. And you don't need to memorize this formula. Basically, you need to know it exists and what it does, and we're going to practice with it. The other formula is the Simpson's diversity index. And they're both just ways of quantifying species diversity, right? We are doing science and we quantify things. So these two indices were developed in order to quantify biodiversity. So that is our biodiversity lecture. Let me know if you have any questions.